All right, hi everybody. Over the past couple weeks, we have been looking at the ways in which our body defends ourselves in a non-specific way. In other words, when we get exposed to the injurious agent, the microorganism that wants to become a disease in our body, so far all we've shown you is ways that our body responds to them in a general way. Like, oh, we're getting infected, now we're going to have to do something about it. That was the innate response, and the adaptive response, which is considerably more effective, we're going to be targeting very specific features of the invading microorganism itself. Specificity is the name of the game when we're talking about this, our adaptive immune response, or our acquired immune response. There are basically 5 things that we need to consider when we talk about adaptive immunity. The first, as I've already said a number of times, is this idea of specificity. Each adaptive immune response is targeted against a single molecular shape unique to the infecting pathogen. So this was the pathogen, and let's say it's got all these little shapes and nooks and crannies on its surface. The adaptive immune system does not target the whole thing, but rather one of the structures that's found on the surface. That's how specific this adaptive response is. It's also inducible. In other words, it's not on all the time. The cells associated with this immune response, like the T cells and the B cells, only become active in response to specific pathogens. So, yes, you may have an adaptive immune cell that's targeted to this nasty pathogen's triangle-shaped structure on its surface, but unless this triangle-shaped structure on its surface makes it into your body, those cells will not get turned on. So there's an inducibility component to this as well. Once one of your B cells or T cells is induced, it has a clinality component. So you may only have 1 or 2 of these anti-pathogen triangle cells in your body, but once induced, these B cells and T cells will just divide like crazy, and that produces tons of clones. Importantly, a 4th feature of the adaptive immune system is unresponsiveness to self. So your cells of your adaptive immune system do not act against normal, healthy cells of your body. Otherwise, if this were to go wrong, and in fact, unfortunately, it can, your immune system will start attacking your own healthy body cells, which would obviously be quite a detriment. And then finally, there's memory. So once you've had a particular pathogen and lived through it, the adaptive immune system retains this information and the ability to respond to that pathogen much faster than the original response, which is why in some cases you're able to get an infection and never have it again for basically the rest of your life. So this is a really weird story because it's very specific, but we really do have the ability to respond to pretty much every living thing that could infect us out there in the world. So most of you haven't had Ebola, for example. You've all heard of Ebola, a big scary virus. It's a terrible virus. Most of you, nor have actually any of your family members or your ancestors ever come into contact with Ebola. And yet I guarantee you have B cells and T cells that are willing to fight the Ebola virus because they have been randomly generated to do so. So even though you've never come into contact with it, there could be an alien virus coming from Mars, who knows? But you actually already have the ability to defend against it, which is really one of the more remarkable stories in biology, I would say. The stars of the show, when we're talking about the adaptive immune response, are called the lymphocytes. You know about these guys already. They originate in the red bone marrow of juvenile long bones and adult flat bones. They are the smallest of the leukocytes, almost the same size as the red blood cell. So you can see compared to a red blood cell, they're actually pretty similar. This is what they look like in a resting state. So basically, you can see a gigantic nucleus with a little bit of cytosol around it. B and C T cells look identical in this form. You wouldn't be able to distinguish between them. But if you were to zoom in and look biochemically on the surfaces of the cell, they would be quite different. And when they become activated, they can look and actually, of course, act quite a bit differently. B cells or B lymphocytes, just quick factoids on them. They arise in the red bone marrow. They mature in the red bone marrow. They are the primary line of defense in the humor. So sometimes we call the B-cell mediated response the humoral response. And sometimes we call it the antibody response or antibody mediated. So humor is just another way of saying the bodily fluids. So this is like if you're in your bloodstream or interstitial fluid, this would be your primary line of defense. And the way in which B cells attack is by producing and releasing antibodies. And we'll talk about what antibodies are doing and what they are in just a moment, to target various pathogens in the body. And that's why the second name of it is antibody-mediated immunity. The other lymphocyte are the T cells, in which you actually have two different ones. There are TH cells, H being a helper, and TC cells, with C being cytotoxic, and we'll talk about the difference between those in a while. These, again, also just like the B cells arise in the red bone marrow, as you know, but you remember from a couple of weeks ago that these are maturing in the thymus, and this word mature, you don't really know what it means yet, but we'll talk about maturation towards the end of this week's work. The function of the T cells is to regulate adaptive immune responses. That's the responsibility of the helper T cells, and attack intracellular pathogens like viruses. That's your cytotoxic T cell. These, when they act, are referred to as the cell-mediated immune responses. So the helper T cell, as we're going to see, can help the cytotoxic T cell do its job, but the helper T cell can actually also go and help the B cell do its job. And in that way, the helper T cell is kind of the center of the story. So in terms of how our body is able to respond, the key structure is called an antigen. The antigen is not a human structure, but rather the pathogen structure. And as I've been saying, I'm trying to talk about them in terms of unique biochemical shapes. In this case, these are the biochemical shapes that your body recognizes as being foreign, as belonging to a foreign invader. And so again, if you looked at this being a bacterium here, if we zoomed in on the surface of the bacterium, you can see, say, maybe some sort of membrane protein with various shapes on its surface. The antigen is the foreign invader, and you can see that there's a large structure on the foreign invader, which is the antigen, and then on the surface of the antigen is all these various molecular shapes. Each one of those molecular shapes is slightly different from the others, and that's actually what's being targeted. That's what's called an epitope. And so even though we're going to be using these terms antigen and epitope pretty interchangeably in this lecture, please know that antigen is a larger structure, and an epitope is actually what's physically being bound to. So how do you, what makes up an antigen? I said it's a part of the pathogenic microorganism, but what makes it easy for our body to identify an antigen? What are we looking for in an antigen in order to appropriately be able to identify foreign structures and get rid of them as quickly as possible? So a couple things to make an antigen easy to identify. One is shape. If something is a very boring shape, very uncurved, very unnuanced, you would expect that to have not very many epitopes on it. Whereas something maybe like this, with a sort of crazy shape, is going to have lots of epitopes on it. So the more complex the shape, the more likely the antigen will be easy to identify as being unique from cells of your own body. Size is important too. What's going to have more epitopes on it? A structure this big or a structure this big? Obviously, the larger you are, the more unique shapes you're going to have and more epitopes you're going to have. And complexity is really what we're talking about, right? Because the more complex, the better. Because the more complexity you have, the more epitopes you'll have, right? Again, you could be this, or you could be something that's huge and has all sorts of different possible nooks and crannies that can be recognized as epitopes / antigens. So here's some examples of common antigens that are found. We've got antigens in the bacterial cell walls, pili, which are bacterial sex structures, a way that they can exchange nucleic acids. We've talked about capsules as a way of bacteria escaping detection from macrophages, or rather avoiding phagocytosis. Bacteria have little tails on them called flagella, which are not the same as the flagella in our cells. But it does do the same thing, causes them to swim. So all of those things, so the pili, which are these little sex structures that are coming out of the bacteria, the cell walls, which are surrounding the bacterial cells, the capsules, which are surrounding the bacteria, and then they've got these proteinaceous tails. Each one of those can be recognized as an antigen because they are exposed to the humors of the body, and then they can be identified. The capsomeres of viruses, so those are the proteins that surround the nucleic acid in the virus that make up the shell of it. We also have various external structures of protists and fungi, mostly in the glycoprotein families, so just a bunch of sugars fixed to protein molecules. And unfortunately, we can also find things that aren't so harmful to us and treat them as antigens too. And these include allergens, such as, unfortunately for me, pollen grain, dander, dust particles, anything that you can be allergic to can be an example of, well, for lack of a better term, an unwanted antigen. There's 3 different classes of antigens that we need to address. The first is called exogenous antigens, the second are called endogenous antigens, and the third are called autoantigens. We've already hopefully explained what an antigen is. Let's start with exogenous antigens first. Exogenous antigens are toxins and other secretions, as well as components of the bacterial cell walls, membranes, flagella, and pili. What does that actually mean, though? Well, it's all the structures that are found on those things that can be used as epitopes. So you can see, like, that can be used as an epitope, the structure or antigen right there. The tail with all these little triangles coming out of it can be used as an antigen. Maybe the toxins that are being secreted by the bacteria are going to have antigens that can be identified. The membrane's going to have proteins in it. The cell wall is going to be unique to bacteria. All of those things can be identified as an antigen. The reason these are called exogenous antigens is because they're on the outside of the pathogen. That needs to be distinguished from the second example, which are called endogenous antigens. And endogenous antigens result from any pathogen that reproduces inside the cells of your body. That's what's going to be producing those endogenous antigens. And our cells of our body can actually detect the internal health of our cells. So ultimately what ends up happening is the endogenous antigens end up getting sent to the surface of the cells. So rather than describe those in more detail right now, I want to skip and go straight to autoantigens. Autoantigens are antigenic molecules that are derived from normal cellular processes. So when your cells are healthy, autoantigens are what will be on the surface of the cells. So these are normal body cell antigens that are produced in your body just through normal metabolic activities. Really what's happening is, in here there's all this cellular activity which is producing various proteins and molecules, and those are what's getting sent to the surface of the cell. And basically, if you were an immune system cell, like a B cell or a T cell or whatever, you would look at these and say, oh, this cell is happy because it's got only autoantigens on its surface. Now imagine this cell over here is unhappy. It's infected. It's got maybe some sort of RNA virus stuck in it. As a result of that, of course, you know that the virus will cause the cell to start producing viral molecules. And so you start making these intracellular viral proteins here, of which, again, just remember, same cases with the autoantigens, a subset of the proteins we make inside of our cells get sent to the outside so our bodies can know what these cells are doing, such that when the body starts producing viral or pathogenic components, those too will also be found on the surface of the cells. So in this way, an infected, unhappy cell can actually start producing viral structures on its surface so that your B cells and your T cells can say, oh, this is an infected cell, we need to do something about it. So the autoantigens ensure that your immune system recognizes normal cells of your body and therefore do not attack them. Endogenous antigens result from a cell producing things it's not supposed to, and therefore making its surface recognizable to B cells and T cells as foreign, so it will become attacked. So this is very common in virus-infected cells. It's also a common way of identifying cancer cells in your body, in that they're going to have weird endogenous antigens on their surface. So the key here for this slide, exogenous antigens are found on the surface of the pathogen and can be directly accessed by our immune system cells. An endogenous antigen is the result of an intracellular infection or defect, which will ultimately result in a change of the antigens on the surface of the host cell to represent those pathogenic proteins that are being made. And autoantigens are just a subset of that, in which a cell is not infected, and the subset of proteins that are sent to the surface are indicative of healthy cellular behavior, and hence won't be targeted by the adaptive immune system. Okay, so that's just a brief overview of the start of the adaptive immune response. In the next lecture, we're going to go over the B cells and T cells in specific detail and talk about what they're doing. And I'll see you then.